Key assessment 6A, question 2. And this is component 2 stuff. And we've got a bit of classification. And it's based on cell structure and biochemical analysis. Here's an electron micrograph, so taken using an electron microscope, of a human cell ingesting a yeast cell. Okay, so it looks like it's enveloping it, taking it in, that sort of thing. Complete the table below. Domain, right? So remember your 3 domains. You've got eukaryote, you've got archaebacteria and prokaryotes and so on. So these are both eukaryotes. Or you could say eukarya if you like. I'm going to stick to good old English. Kingdom. Again, you can go Animalia, but I'm just going to write animal. You could write Animalia in the Latin, and we're going to have fungi there. And cellular basis. So tell us something about the cell structure to separate these into different kingdoms. So things you can't say, well, they're both, nuclei. Well, they've both got nuclei. They've both got mitochondria, and both got cell membranes. Both got a load of other stuff in. That's exactly identical. But a difference here, one of the most obvious differences, is in the cell wall. The human does not have a cell wall, and the yeast does have a cell wall, and it's made of chitin. So that's what we need. So no cell wall and cell wall of chitin. So a nice clear difference between them. Calculate the magnification of the electron micrograph above. So what information do we have? We have got a scale bar here. So we've got a scale bar. This is the information that you need. I'm going to put my ruler on this scale bar. I'm not going to measure this. I've got no idea how big that is. There's all sorts of answers measuring all sorts of things. This is how you do it. I'm going to put my trusty ruler on this scale bar, and I'm going to read how big that scale bar is. That scale bar is, on my ruler, 11 millimeters. So that is 11 millimeters. And now my calculation is, so my image size is 11, my actual size is 1 micrometers. I need to put these into the same unit, so my image size is 11 millimeters, and I'm going to convert this into the same units as the one I'm given. So that's 11,000 micrometers. My actual size is 1 micrometers. And so my calculation is, for magnification, is how many of my 1 micrometers go into my 11,000 micrometers. So my magnification is my 11,000 divided by my 1. And when I do that on my calculator, anything divided by 1 is itself. So my magnification is 11,000. The mark scheme says 12,000 and a few other crazy answers, but this picture isn't quite the same size as it was originally. It's a bit reduced, so you can't really say 12. Part 3. Explain why the process shown in the electron micrograph is an example of holozoic nutrition. So this is just remembering AO1. What is holozoic nutrition? It's when something is ingested, and it even mentions ingesting here in the question. So digestion is internal or inside the other organism or cell or whatever. In this case it's intracellular, which means within the cell. I'm going to put intracellular. Describe the digestion of the yeast cell, including the role of the Golgi body. So even though this is KA2, which is relating your knowledge to this diagram, it's really just remembering some stuff. So what does the Golgi do? So Golgi, so it asks us about the Golgi body. So modifies proteins, processes proteins, but what we're talking about digestion here. So Golgi body, it probably modifies enzymes or makes the enzymes, that sort of thing. So Golgi body produces or modifies proteins into enzymes. If you said makes enzymes from proteins, or folds proteins into enzymes, any of those sorts of ideas there. This will also package these enzymes into lysosomes. Okay, so we've got Golgi is produced in lysosomes, and then we want the actual digestion. What's gonna actually happen? So we'll have this yeast cell. This yeast cell has been engulfed by this. So been engulfed by this body cell, and eventually it'll form a vesicle inside. So that's my vesicle, that's my yeast, and this is the human cell. And so we've got our enzymes being made on rough endoplasmic reticulum, we've got our Golgi there, and we've got vesicles called lysosomes that will move towards this, fuse with this, release the hydrolytic enzymes, and then digest this yeast. So we just need to add that onto our story. So lysosomes fuse with the vesicle containing the yeast and release the enzymes. So we've got the Golgi, packaging, modifying these enzymes into a lysosome that will then fuse with the vesicle containing the yeast, or the phagocytotic vesicle, release these hydrolytic enzymes which will then digest the yeast. And there's 2 marks in there somewhere for that story. Now we're talking about viruses. Similar to other organisms, they contain nucleic acid and can reproduce, but not on their own. They are not considered to be cells. And here's another electron micrograph. Gives us our magnification. We're going to have to do another calculation again in a minute, aren't we? Describe the structure of a virus and explain why it's not considered to be a cell. Hopefully pretty straightforward. So it's a molecule of nucleic acid, e.g. DNA or RNA, and that's surrounded by a protein coat called a capsid. So that's our first mark. That's the structure of a virus. And now we need to go, well, why is it not considered to be a cell? And that's going to be the rest of our answer down there. So this is AO2, applying our knowledge of cell structure and virus structure to this argument. So virus does not have a membrane. So if I ever think of a cell, I think, well, cell membrane. Or if I think of a cell cytoplasm, that sort of thing. Or organelles, possibly, even though bacteria don't have them. So I'm going to go for membrane or cytoplasm as another suggestion for my second mark there. Here's the calculation. Calculate the actual length of this bacteriophage. Show you working, express your answer. So I'm going to show our working and express the answer to 2 significant figures and suitable units. So there's 3 marks here. So we don't want to blow this 3 marks by giving it in the wrong fashion. So we're going to have to. So our magnification is 135,000. Our image size is trusty ruler time. Our image size, put that on. I'm going to have 17 millimeters for that. 17 millimeters. And our actual size is what we are calculating. So our calculation is just going to be, we're going to take the size it is on the paper and it's going to be 135,000 times smaller, so we're going to divide it. But our units, well, if we're divided by that huge number there, then I'm going to get rid of millimeters and certainly use micrometers instead of millimeters. So that's going to be 17,000 micrometers. And that's probably going to give us a more sensible. We might have to go to nanometers. Let's see what the trusty calculator gives us. So our actual is the image size divided by our magnification. So let's show you working. So that's going to be 17,000 divided by 135,000. Not 0.125. So my answer is 0.1259 micrometers. And now it wants 2 significant figures. So I'm gonna 0.13. So I'm gonna 2 significant figures there. That's the 1, 2, and the next one's 5. So I'm gonna have to round it up. Micrometers, and that's what I'm going to go for. I think that's fairly sensible. 0.13 micrometers. You might have I'm going to express it in nanometers, so I'm going to times that by 1,000. So if nanometers, then that's 125.9 nanometers and 130, 2 significant figures, 130 nanometers. But I think micrometers is alright for this today.